Hi guys, and welcome to Health Insight Hub, where we discuss important topics about health and science. So today we are dealing with the third part of the cellular biology series, which is gonna be about the electron microscope, its types, and the most commonly used methods to prepare sections so they can be seen on the electron microscope. So let's start. For the electron microscope, we have two types. We have the transmission electron microscope and the scanning electron microscope. So for the transmission electron microscope, TEM, uses electrons which move in a tube where a very high vacuum reigns. The flow of electrons is provided by a cathode, tungsten filaments heated by an electric current. The electrons are then accelerated by an anode, which makes it possible to establish a potential difference of 50,000 to 100,000 volts for a normal microscope, and of 2 to 3 million volts for a high voltage microscope. So, how does it work? An electron source at the top of the microscope emits electrons that travel through a vacuum in the column of the microscope. Electromagnetic lenses are used to focus the electrons into a very thin beam, and this is then directed through the specimen of interest. The electrons passing through the specimen then impact on a detector. We are still in the how does it work slide. So now we are talking about the final results, what we see in the image. So for traditional bright field imaging relies on incident electrons being scattered and disappearing from the beam depending upon the compositional density and crystal orientation of the sample. So the intensity of unscattered electrons gives rise to a shadow image of the specimen with different parts of a specimen displayed in a varied darkness according to density. Then here are some facts about the transmission electron microscope. Separating power close to a nanometer, an analytical technique used to visualize the smallest structures in matter. Observation of cellular ultrastructure. Due to the low resolving power of electrons, the sections must be very thin, 40 to 60 nanometer thick. And to have these thin sections that are 40 to 60 nanometers thick, we are going to use an ultra microtome. So ultra thin sections are made using an ultra microtome and are collected on grids. For grids, we have two types of grids. It's not a big deal. In the form of a fence with a hole in the middle, and it's called single hole fence grid. As we have seen in the previous videos, that for a structure to be seen in a microscope we usually color it. So for the electronic microscope, or for the transmission electron microscope, we use the negative staining. Negative staining is an excellent way to study very fine structures, molecules, viruses. Here are some examples of these fine structures: ribosomes, myosine and tubules. Myosine and tubules. This technique makes it possible to contrast not the cellular structure but the space that surrounds it. So here are some steps for the negative staining. Isolation of the substances to be studied. Put them in solution with phosphotagnistic acid, which they do not absorb. We deposit this solution on the collagen membrane grid, single hole grid. We'll let the grid dry. After drying, the phosphotagnistic acid precisely delineates the structures to be studied. Wherever it is, electrons cannot pass through. On the other hand, electrons pass through the structures that we want to observe. We therefore obtain negative images. Here are the freeze fracture technique. So for the freeze fracture technique is a very easy technique, really easy to understand. We use it to cut and to fracture specimens, to have small sections. So for cryostripping, it's almost the same thing as the freeze fracture technique, except that we increase the precision of the reliefs. In fact, in this case, we carry out a sublimation. So for the sublimation, what is sublimation? Sublimation is when we pass from a state to another state. So from the solid states to the vapor states. Let's give an example of water. So when we have ice cubes, we are going to go from ice cubes to transform water from ice cubes to vapor without going through the liquid phase. This is the thing. So sublimation of the ice under vacuum before adding the heavy metal salt. By removing the water stuck between the proteins, we see more relief. The freeze fracture technique. The freeze fracture technique consists of freezing and then physically breaking apart, fracturing a biological sample and visualizing the exposed structural detail of the fracture, by vacuum deposition of platinum carbon to make a replica for examination in the transmission electron microscope. So to simplify this freeze fracture technique, let me give you an example. So imagine that we have a cell and we want to apply this technique on this cell, and we want to see it on the electron microscope. So we are going to fracture the cell into two pieces. Then we're gonna deposit a platinum and carbon layer on the cell, on the two parts of the cell. Then we are going to put them dry for a moment, then take the layer. For sure the layer is going to take the structure of the cell, so we are going to have a we're gonna have the form of the cell on that layer. Then we are going to discover it and see it on the electron microscope. Here is the thing. So to make a replica for examination, we're gonna have a replica for examination in the transmission electron microscope. This is the thing. So here are some steps that we should follow to have a replica, to observe it on the electron microscope. So for the first step is fabric placed on a support. Second thing is freezing. Then the tissue is placed in a vacuum chamber. Frozen tissue is fractured. Fabric surface, shading of the released surface. Replica of the surface isolated by enzymatic digestion of the tissue. The photograph of the replica showing the envelope of the nucleus and its pores. This is the thing. Now let's move on to the second type of electron microscopes. Now we have the electronic scanning microscope. So the scanning electron microscope, or electronic scanning microscope, allows observation of the surface of the sample. The electrons emitted by the cathode will scan the surface of the sample. The surface of the object to be scanned is previously covered with a metallic layer. And we have here a 3D view of the specimen. So here are the steps to prepare a biological sample. Fixing, dehydration, cut into thick slices, not as the transmission electron microscope. We have very fine structures, 40 to 60 nanometers. Here we have thick slices. Gluing on a metal support plot, and metallization of samples to make the samples conductive. It is necessary to cover them with a thin layer of metal, and we usually use the gold as a metal. So we have finished the third part of the biological cellular series. We still have just one part to deal with. It's the fourth part. I'm gonna upload it in 3 days, inshallah. So stay tuned for the fourth part. I hope you've enjoyed the video. You've learned a lot about it. It's a really interesting topic to search. You can go and search more about things. If you have any question, just feel free to ask me in the section, in the comment section. And stay healthy, stay tuned. and stay informed. Bye, guys.